In this video, I will be going over the Trypan Blue Protocol by using a hemocytometer. Make sure that you have glass pipettes, Trypsin, PBS, Trypan Blue, pipette tips for the P1000, an automatic pipette, and the P1000. Before counting any cells, the first thing you'll want to do is make sure that you have 300 microliters of Trypan Blue in a centrifuge tube. These are in sterile individual packets on the lab bench behind me. And on the pipette, it'll read 030. Remember, you have to add another 0 on the P1000, and that'll show you that you have 300 microliters. Once you have everything set up, you can take out your cells from yesterday's experiment, and you'll take a look at them underneath the microscope to make sure that they look good, that you can see them, that they're all there. Your cells will look more abundant than what you see here. As stated previously in the previous videos, there was a lower cell count. I didn't plate that many cells, but you should see a lot more cells on your dish. Now, once you see your cells, you can go into your fume hood, and you are going to take the following steps. You're going to aspirate the medium that the cells are sitting in. Now you're going to rinse your cells with PBS. You can take the P1000 and pipette a few squirts of PBS in there and then aspirate it. Now you're going to make sure that you have your automatic pipette, and you are going to add 1 milliliter of trypsin to your dish with the cells in it. Place the dish in the incubator for 4 minutes, and then you can pull it out. 0.5 milliliters of PBS. So you could use the automatic pipette, or you could use the P1000. Just make sure that your measurement is correct on the side, that you have 500 microliters if you're going to use the P1000. I'm going to shake the dish a little bit to mix up the cells with the PBS, and then you're going to take the P1000 and use a sterile tip to mix the cells up a little bit more. When you're mixing the cells up, you are only going down to the first stop on the pipette to prevent any backup into the actual pipette device. Once you're done mixing the cells, you are going to take 300 microliters of the cells and add it into your Trypan Blue. Invert the tube a few times to mix the cells with the Trypan Blue, and then place a centrifuge tube in the bead bath completely submerged for 3 minutes. Now you can take the cells out, and you can use the hemocytometer in order to count how many blue cells you have and how many white cells you have. Blue cells are going to be the dead cells, white cells will be the living cells. Similar to the previous video for cell counting, you will have to take either the P20 or P200 in order to pipette your Trypan Blue and cell suspension mixture into the hemocytometer. You need to do this at an angle. And you can place the hemocytometer on the microscope, and you are going to look for those same grids that you would when you're counting cells to plate them. When you look at the grid, you should see mostly white cells and some blue cells. In this case, the cell count is low, so you see a few white cells. However, when you're looking at them, you'll definitely see a lot more cells, and you'll be able to count all of them. You should be counting the cells on the hemocytometer in A, B, C, D, and E, and then make sure you write down how many blue cells you have and how many white cells you have, because you'll be using that for a calculation to calculate cell viability. Make sure to clean the hemocytometer and the slide. Use distilled water and make sure that everything is dry and clean, because Trypan Blue does stain. When you're ready to count cell viability, you will need to take the number of blue cells over the total number of cells, so blue plus white. Divide that subtract 1 from that number, multiply times 100, and you will get your percent of viable cells, so cells that are living from that dish. And then you'll know how well your experiment went based off of this outcome. If you are testing multiple dishes and you're repeating the same steps for each dish, so let's say you have 3 dishes and you do this 3 separate times, you will have to count cell viability and do this protocol 3 separate times, because each dish could have different results.